Welcome to this biochemistry lecture on RNA technologies. So first we're going to discuss RNA as a drug target, and there's several problems that exist with RNA as a therapeutic target, mainly the size of the molecule, the complexity, and the intrinsic flexibility of these molecules. And so we can attack this in RNA as a drug target in multiple different ways. We can basically bind it and inactivate it, and that's the main way. Or we can, as a drug target, that's the main way we're going to do it. And you're going to do this by either proteins or other RNA. And RNA does perform a variety of functions and roles, and those are important and can be very important in drug targeting. But you have to know the specific ligand interactions and how those are going to interact, how your drug is going to interact with the RNA, and how that's going to inactivate it. Basically, you need to know the mechanism of action. So you're starting to see things like this lipid nanoparticle that has RNA in the middle, and nucleic acids in the middle, and they've committed a cartoon here where you have very large polymers, and in particular, you're delivering RNA to a cell in order to knock down other RNA, and so you're starting to see this. Companies like Moderna are using these technologies to facilitate delivery of other RNA to knock down RNA as a drug target. You're also starting to see RNA used as a vaccine very commonly, because what you do is you make a few modifications to RNA to stabilize it. You deliver it as a vaccine, and that mRNA gets translated into a protein, and the cool part is that it has the conformation that the cell would have versus, if you produce it synthetically in something like E. coli or even other cell lines, you don't necessarily know if the conformation is going to be the same. Well, if you deliver mRNA to a site, you know that the conformation will be very similar generally. So this is the kind of study that you would do. You'd put a lipid nanoparticle with mRNA to stabilize it, and you'd have a 5' cap, your UTR, your poly A tail, all that kind of stuff. In this situation, it's a Zika virus, so you're going to put in PRM, which is one of the proteins, E, which is one of the proteins that's on the envelope, and they're fusion proteins, and you deliver that, and you give it to. You can test it in mice or primates, and you can see how it's going to be used. You can see how effective it is, efficacious and safe it is as a vaccine. And you avoid all the complexities of making protein. Lastly, we use RNAi, sometimes called RNA silencing. It's very commonly used to see for transient effects, and basically you take the complementary strand of an RNA and you deliver it to a cell that knocks it down and knocks down gene expression, and so you can knock down any kind of protein you would like, and that can obviously improve or cause problems. So you can use it to characterize a phenotype of a gene silenced, or you can use it as a therapeutic target. This was emerging about 15 years ago, and it's a very transient effect, though, so you're not going to use it for a long term. It's going to transiently knock down gene expression. It's going to transiently knock down RNA production, and basically the concept is that a complementary strand of DNA is again used to silence the translation of a product. That's what we have today. Appreciate you listening. RNA technologies definitely are going to be improving, and they're going to be challenged. They're going to challenge some of the more traditional drug delivery mechanisms over time. And so keep an eye on this field as it goes forward. I look forward to talking to you later.